Kicked off. Perfect. Our first speaker, Kevin Liu. Are you ready to go?
Yes, I am.
All right, take it away.
Great. Thank you so much, April. Yes, so I'm Kevin Liu from the Department of Computer Science and Engineering and the Ecology, Evolution, and Behavior program at Michigan State University. Thanks to all the conference organizers for putting together this wonderful virtual conference. Yeah, I'm here to talk today about an issue with co-phylogenetic reconstruction, where upstream phylogenetic tree estimation error has some serious impacts, as we'll see. Before I begin, I'd like to just acknowledge all of the contributors to this work, which include my PhD student, Julia Zhang, who did all the real work, as well as some of my other collaborators at Michigan State. So just as a phylogeny encodes the evolutionary relationships amongst a set of taxa, as in this example for a species phylogeny, a cophylogeny encodes the coevolutionary relationships amongst a set of co-evolved taxa. For example, if you have a pair of host taxa and symbiont taxa, you can represent the present day relationships here on the left using a tanglegram, where you've got different associations between sampled host and symbiont taxa. But then going backwards in time, you can try to reconstruct all of the coevolutionary events that led up to those present day associations that you see. So these can include things like co-speciation, as well as other events such as duplication, loss, and host switching. And so this is an example of a species cophylogeny on the right. One more piece of terminology is the present day associations and the two trees can be represented using a tanglegram, which is typically the input for species cophylogeny reconstruction. And so there's lots of different applications where cophylogenies are of interest. So if you're trying to study the origins of these coevolutionary relationships, that's one example. Trying to study more generally coevolution as a process in the tree of life, and lots of other applications as well. So for example, epidemiology, et cetera, are situations in which cophylogenies are of interest. And just like with phylogenies, cophylogenies are reconstructed using computational methods, typically by analyzing biomolecular sequence data and other types of data. And these are broadly categorized into two classes. First off, you have these so-called cophylogenetic reconciliation methods, which take in, as I said, these tanglegrams, which are really a pair of trees in the simplest case, a host and symbiont tree, in addition to the present-day host-symbiont associations for the taxa. And these try to reconstruct the historical coevolutionary events in the form of the cophylogeny, typically under an event-based model. And so some examples include Empress, Coripa, Tail, and others. In addition to this, there are so-called global fit methods, which really are trying to just at a more high level test a hypothesis about coevolution, as well as other high-level assessments. Will mostly be, or actually entirely be focused on these cophylogenetic reconciliation methods today. And so these methods really operate within a larger pipeline, just like with phylogenetic reconstruction. First off, typically a multiple sequence alignment is produced from the underlying sequence data, from which then trees are estimated as in a typical phylogenetic analysis. Then a tanglegram is formed, so these trees are coupled with those present-day associations, after which the reconciliation is performed to try to reconstruct these coevolutionary events and in the form of the cophylogeny itself. And so, as you can see in this pipeline, there's basically this practical assumption, which is that upstream estimates are used directly as inputs for downstream analysis. And so when you're doing this, it can be the case that upstream error can propagate, upstream estimation error can propagate throughout the pipeline. And so in particular here in this stage of tree estimation, it's well understood that this is not a perfect process. There's lots of different opportunities for error to creep in. And so the question that we looked at was, to what extent does this impact downstream cophylogenetic reconstruction, this issue about upstream tree reconstruction quality? So like I just mentioned, it's well understood that this is an important issue to consider. And so there's lots of different factors that can cause this, in addition to just the problem of phylogenetic tree reconstruction being computationally difficult. And so the heuristics that are utilized typically can introduce error. There's also the opportunity for model misspecification, alignment error, sequencing error, and other sources of error. And so in this particular context, we just wish to not only establish that upstream tree estimation error can impact downstream co-phylogenetic reconciliation, but also to quantify these impacts in this particular study. So this is essentially what we set out to do using a series of experiments, as well as to provide practical guidance. Today, I'm just going to show you a few highlights in the time that I have. And so the first half of the study really looks at a bunch of simulation experiments to try to quantify these impacts. And for the simulations, here's just a really high-level overview of kind of how that works. You have to first off get a bunch of model trees. And the way in which we did this was essentially we went back through the literature. We got a set of cophylogenetic studies, got their data, reconstructed trees, and these served essentially as parametric models for our simulations. And then we actually had a few different approaches to actually conduct the simulation procedures. I'm going to highlight just one today, the so-called mixed procedure, from which we basically took not only the reconstructed trees, but also the co-phylogenies that were reconstructed by those studies. These essentially served as our ground truth and as parametric model for simulations, from which we basically utilized kind of a simulation software to simulate sequence evolution along those trees and get synthetic data. And essentially, we compared estimates from those synthetic data using that pipeline I just showed you and compared that against the original empirical estimates. That's one approach. We actually had another approach that was fully in silico, where we were using completely random trees. We're generating actually ground truth cophylogeny, synthetic cophylogenies, and model trees using a particular software package, TreeDuckin, from the Heath Lab and others, and other software packages as well for the fully in silico approach. But I'll mostly just be focused on the so-called mixed approach. And then to actually do the comparisons of the estimates versus either the reference or ground truth, we used the Robinson-Folds distance to compare the trees based on their topological differences. For the cophylogenies, we used a similar calculation from Wesecki et al., which essentially looked at the proportion of coevolutionary events in one of the cophylogenies and not the other. And so here, just at a very high level, is a bunch of the different simulation conditions that we dealt with. And so there's really 6 in the mixed simulation procedures, covering a bunch of different studies of things of gopher and lice, and other kind of host symbiont studies from past literature. And they involved kind of a range of data set sizes, both in terms of number of taxa, sequence length, and evolutionary divergence. That's basically the main takeaway from this particular table. There's just a series of simulation conditions used for our study. Here quickly are the results. Let me orient you into what we're seeing here. Basically, on the bottom horizontal axis, I have the topological error. So this is comparing the reconstructed trees against the reference trees for our simulation study. Here are the mixed simulation experiments. Kind of left here is more accurate. Remember, this is error. And as you move rightward along the horizontal axis, it's increasing in error. The vertical axis is this topological comparison of cophylogenies. This, on the other hand, is an accuracy calculation. So the higher you are, so up here on the top is more similar cophylogenies, estimated versus reference. As you go down is more dissimilar, okay? So you kind of have to orient yourself here. And essentially, each data point is an individual replicate for the estimation errors that you're seeing of the estimates versus reference. And so all we can see is, generally speaking, the main takeaway from this is, in addition to the scatter plot, you're seeing a regression line, a simple regression line, in addition to kind of a confidence band. And again, the main takeaway that you're seeing is generally, you can kind of eyeball it here, as you have more accurate trees here on the left of the horizontal axis as input into that pipeline, you're essentially getting more accurate, more similar cophylogenies, reference versus estimated. And as you kind of get less and less accurate trees, you get less and less accurate cophylogenies as well. For this particular simulation condition being mixed gopher, and it was a statistically significant result here. And very, very quickly, we essentially saw roughly the similar outcome across all of these other conditions as well. And so some of the differences that you do see are due to just simply the different characteristics of the simulation conditions themselves. Data set size and evolutionary divergence playing a key role. So in the paper that was published for this study, we kind of delve into this a little bit more. Briefly, we also conducted an empirical study as a companion to the simulation study. I'm only going to talk about one of the data set analyses that we looked at, which involves soil-associated fungi and their bacterial endosymbionts. One being for this very kind of early diverging soil fungus from Mortierella, from which we had a resequenced whole genome sequence data that we analyzed. We utilized a fairly standard pipeline for analyzing that data set, both for phylogenetic tree reconstruction as well as co-phylogenetic reconstruction. And because we don't have reference or ground truth in this particular instance, instead what we set out to assess was, okay, if we have a set of different methods for upstream tree reconstruction, some of which potentially are less accurate versus others, then we can study kind of its effects upon reproducibility and disagreement on downstream co-phylogenetic reconstruction. That's essentially why we made different choices for each of these reconstruction stages within that larger pipeline. And so once you do that, then you can kind of compare all of the different estimates, trees and co-phylogenies, using the same approaches that we utilized in the simulation experiments. And the results are reported similarly as well. Here are the results presented in basically a similar fashion, although, so for example, the horizontal axis here, same kind of layout as in the previous figures that I just showed you, vertical axis, same as well. So on again, remember that the horizontal axis is topological, disagreement here. More agreement here is on the left, less on the right. The co-phylogenetic agreement here is this vertical axis, more agreement on the top, less agreement on the bottom. And you're also seeing this regression line with the band as shown with the scatter plot. Layout similar. The only kind of caveat/difference is, remember, no ground truth, no reference here. We're just looking at disagreements amongst the downstream estimates where the pipelines differ in terms of their kind of known estimation error. That's essentially what's happening. And so the takeaway, again, kind of similar. Where you have less accurate methods upstream, essentially what we saw was more disagreement results kind of downstream for co-phylogenetic reconstruction. That's the main takeaway here. So it is a consistent result, although it is a somewhat different setup because obviously this is an empirical data set rather than a simulation experiment. Okay, in conclusion, what I presented is just a portion of the study where we've quantified the impacts of upstream tree reconstruction error on downstream co-phylogenetic reconstruction. And we conducted a series of experiments using both theoretical and in silico simulations as well as empirical data set analyses where we really did quantify this impact. And we saw there was a significant and large impact with basically this negative relationship in all cases. And so our recommendations are generally speaking that, well, it would be great if we had new methods that essentially tried to better account for upstream tree reconstruction error. There are statistical methods that try to partially do this so far. Tail is a good example, which was in fact studied in this particular study. We had experiments in the the supplementary appendix that talked about this. On the other hand, a full solution really doesn't exist. So there really does need to be more methodological research in this direction. In the interim, kind of a workaround is, generally speaking, people should try to adopt more intensive, more accurate species tree reconstruction as a best practice for that upstream tree reconstruction stage, such that you have less error to worry about when you're trying to do the downstream analysis. Yeah, I'd like to acknowledge the funding agencies that supported this work. And that's basically it. Here's some links. If you're interested in more information, the paper associated with this was published previously. You'll find that link as well on these websites. Thanks, everyone.
Excellent. For one question, anybody has a question, you're welcome to put your hand up and I will unmute you. That was a really wonderful talk, Kevin. I'm sorry I had some internet connection difficulties during it because I would have loved to see all of your results. Excellent. Well, it is 9:15, which means it is time to move on to our next presenter. Sean, I think you are in the room.
Can everyone hear me? All right.
Yes, we can
Okay.
hear you. Do you want to go ahead and put your slides up?
Okay. All right. So let's get started. Hello, everyone. My name is Zixuan Chen, and I also go by Sean. I am a statistical PhD in the Ohio State University. So today I will show you a new method using adjusted composite likelihood for speciation time estimation. So first, this is our problem in species level phylogenetic inference. We want to use multi-locus data or SNPs to estimate a species tree for some closely related species under the multi-species coalescent model. And we want to do it quickly and accurately. Under the multi-species coalescent model, we assume the data generating mechanism as shown in this figure. Within the species tree, so the gene trees at different loci may have different evolutionary histories because of the randomness of gene evolution. We use the coalescent process denoted in the blue arrow to represent this to model this variation. And then for each gene, the mutation process is modeled by standard nucleotide substitution models, for example, the Jukes-Cantor model. And because gene trees are not directly observed, we need to integrate out the gene trees when we want to estimate a species tree directly from the sequence data using a likelihood-based approach. So under the multi-species coalescent model, the marginal species tree likelihood given the sequence data integrates over all the possible gene trees that may lie within the species tree. However, as the number of species increases, the number of all possible gene trees increases explosively, as you can see in the table. So this makes the calculation of species tree likelihood nearly impossible for trees with more than 4 taxa. So assuming all sites are unlinked, Schiffman and Kubalko derived simplified site pattern probabilities for species quartet under the Jukes-Cantor model. Under this model, the likelihood of species quartet can be easily computed using the site pattern probabilities. Then what about the larger trees? Well, for larger trees, we can use the composite likelihood framework. So under mild regularity conditions, composite likelihood estimators are known to have good statistical properties, such as statistical consistency and asymptotic normality. So for example, this 5 taxon tree can be broken down into 5 quartets, subtrees, by removing one species at a time. And then we can compute the likelihood of each quartet using the species quartet site pattern probabilities we introduced before. And the product of these 5 quartet likelihoods is the composite likelihood of the 5 taxon tree. So here, let me quickly go through the technical details, and then we can resume back to the big pictures. Here, QJ is the topology of species quartet subtree. Tau j and theta are the species tree parameters related to the quartet QJ. And it has been shown in Schiffman and Kubalko 2015 that quartets will have 15 site pattern categories. So we let Z1 to Z15 to denote the observed 15 category count, and P1 to P15 to denote the theoretical 15 category probabilities derived by Schiffman and Kubalko. Then the quartet likelihood of the is basically a kernel of the 15 category multinomial distribution. And then the composite log likelihood of the 5 taxon tree is the summation of the quartet log likelihoods. So composite likelihood is fast to compute, but an adjustment to the composite likelihood is needed for Bayesian inference. An unadjusted composite likelihood results in an overly concentrated posterior because information is used repeatedly. Lipotyphlan et al. In 2012 proposed to use a curvature adjustment to adjust the composite likelihood to achieve an accurate Bayesian inference. Here, accurate means that the nominal levels of credible interval should match its empirical coverage. So we have done a simulation study to show the performance of the composite likelihood adjustment. First, we fix a 15 taxon species tree and simulate 1 million unlinked sites with 1 lineage per species using PopStar. Second, we used Markov chain Monte Carlo to draw posterior from the unadjusted composite likelihood versus from the adjusted composite likelihood. Third, we construct credible intervals at different levels, ranging from 1% to 99%, and count whether the interval contains the true value. And fourth, we repeat step 1 to 3 for 100 simulation runs and calculate the empirical coverage of the credible intervals at each nominal level. And this is our 15 taxon species tree that is used in this simulation study. Speciation times are labeled numerically by post-order traversal, and we will have 14 speciation times, tau 1 to tau 14, and 1 population size parameter, theta. And to save space, we only show the results for speciation times, tau 1 to tau 6. So this is a posterior comparison plot. As we have mentioned earlier, the posteriors from the unadjusted composite posterior will have a spikier distribution. You can see in the plots. So we would expect the empirical coverage of the unadjusted composite posterior to be lower and the empirical coverage of the adjusted composite posterior to match its nominal level. And you can see the posterior from the adjusted composite likelihood is much flatter than the unadjusted composite posterior. And finally, this is we call a calibration plot that can show the performance of the method in terms of coverage of the credible intervals. So the ideal calibration curve would be the gray 45-degree diagonal line, which means that the empirical coverage of the credible interval matches exactly the nominal level. And the pink dashed lines denotes the unadjusted composite posterior, and they are much below the gray 45-degree diagonal line, which means that the empirical coverages are much lower than their corresponding nominal level. This is because the unadjusted composite posterior are spiky and overly concentrated. And the green dotted curves will correspond to the adjusted composite posterior, and they are very close to the gray diagonal line, which means that the curvature adjustment works. So we conclude that the composite likelihood adjustment provides accurate Bayesian inference of speciation times along a fixed species tree. Moreover, each simulation run only takes about 40 hours for the comparison between the unadjusted composite likelihood and the adjusted composite likelihood. So we can expect for about 20 hours runtime for a 15 taxon tree inference, which is really fast for a fully Bayesian methods in our fields. Our future work will extend the method to estimate the species tree topology using MCMC. Also, we want to incorporate additional substitution models, such as being able to use SNP data or allow rate variations across sites. This has been implemented under the frequentist composite likelihood paradigm in Kubaco et al. 2025. And lastly, we want to use this composite likelihood framework for hypothesis testing. And we are expecting to submit our paper later this year if you are interested in our work. All right, thank you for listening.
Great, thank you. We have a couple minutes for questions.
We have a message in the chat from April, and she says, did you say that the model was a Jukes-Cantor model?
Yes, I did. It's under the Jukes-Cantor model because the site pattern probabilities that are used are under the Jukes-Cantor model here. So we are using this simplified set pattern probabilities of the 4 taxon species tree to derive the composite likelihood. Yes.
So she has to follow up. So then this model could be expanded to morphology then?
This method definitely can expand to morphology. I mean the method of composite likelihood. But for our method, since we are using the data from the sequence, the DNA sequences, I'm not sure whether, yeah, I don't think the site pattern probability can be directly converted to the morphology data. Yeah. Thank you for the question. Thank you.
Awesome. Great job. We can go ahead and get the next speaker since we have about a minute until their talk to share. Thank you again, Sean.
Okay, I think it's me.
I'm going to try this again. My internet's been bad. Yes, it is you next. Daniel, do you want to try getting your slides up?
Yes. Okay.
If that looks, I'm able to see things. So that's.
Okay, is everything all right?
I think we're good.
Okay.
Yeah. Just I'm gonna have you wait a couple more seconds that we stay on the exact half hour. All right, go ahead, Daniel.
Okay, thank you very much. Hi everyone, my name is Daniel Nakamura. I'm a PhD student from the University of São Paulo, Brazil, and also the American Museum of Natural History in New York. Today, I would like to talk about the impact of phenomic data on total evidence analysis dominated by DNA sequences. So, phenomic characters, such as those related with external morphology, osteology, bioacoustics, were the primary source of evidence of phylogenetic analysis in the first decades of cladistics. However, with the advent of PCR and central sequencing, we have now a dominance of molecular sequences as the primary source of evidence, at least for extant taxa. This trend has been reinforced since the invention of high-throughput DNA sequencing in the 2000s due to the declining costs and the increasing speed for generation of large molecular datasets, as opposed to the time-intensive, the taxon specialized collection of morphological data. Although we have now cases of successful sequencing of ancient DNA from old samples, in general, the use of phenomic evidence is still the only source of evidence to include fossils. This is a huge advantage for phenomics, but their low number, typically from dozens to thousands, relative to molecular characters, which typically thousands to millions, has sparked diverging opinions about the value of phenomics in part of the scientific community. On one hand, several authors have challenged the relevance and the reliability of morphology to impact topologies, and only map morphology a posteriori on molecular trees using phylogenetic comparative methods. In contrast, many authors advocate in favor of a total evidence approach, which is a name. This approach was, the name of this approach was given by Arnold Klug, in which phenomics participates of hypothesis testing. And the reason is simple. You don't need to partition your analysis or exclude morphological data, because the more empirical evidence you have, the more severe is your hypothesis testing, according to the total evidence approach. Clade support is typically assessed using two kinds of metrics. The first one are those proportional to optimality, like the Goodman-Bremer, and the second one are those that rely on resampling techniques, like bootstrap and jackknife. Resampling metrics are more popular nowadays in phylogenetics, and specifically, high values of bootstrap are often interpreted as indicator of either a high accuracy, that is a high probability of a clade being true, which is a naive approach because we don't know the true phylogeny. And another interpretation is a high repeatability, which is the probability of a clade being repeatedly recovered again, given resampling or perturbation of your characters or the addition of new data in the future. However, we don't know exactly the relationship between bootstrap and the addition of phenomic data. We don't know if the addition of phenomic data will increase, decrease, or don't change at all the bootstrap values. And we also don't know if we can use the bootstrap from molecular clades to predict the occurrence, the presence of the same clades in the total evidence trees when we have the addition of phenomic data. So here, we used 52 empirical data sets comprising 9 to 500 leaves, 13 to 4,000 phenomic characters, and 500 to 2 million molecular characters. So we have here data sets with different size here. And for the phylogenetic analysis, we used parsimony and maximum likelihood. For parsimony, we used TNT. We performed molecular and total evidence analysis. For likelihood, we used IQ3. We performed the molecular and total evidence analysis, but for the total evidence analysis, we used both the correction and we found the correction from Lewis. This correction is necessary because we usually only sample morphological characters for that have variation. People don't sample usually characters without variation, and for molecular sequences, we don't know how we cannot assess this variation a priori. So we sample everything usually. And for tree comparisons, we computed the number of shared clades, the number of unique clades, some topological metrics, topological distance metrics like the number of SPR moves, Robson folds, cluster information distance. And using all this information, we answered 5 questions, 3 of them related with the impact of phenomic data on topology and 2 questions about support. I will specify each one of these questions in the next slides. Oh, and we also wrote an R package called Arnold, in which we can automatically, given as input trees, 2 input trees, we can automatically prepare a data frame with the matching nodes and the support values and the composition of descendants from that node. Everything can be used for statistical analysis. Okay, so the question 1 is, are topologies impacted by the addition of phenomic evidence? Our results indicated that yes, the incongruences between molecular and total evidence trees were found in 90% of the data sets. And when we performed some statistical analysis using the Wilcoxon tests, we also had the result that the incongruence between molecular and total evidence trees tends to be significantly different from 0. The question 2 is, are molecular and total evidence incongruences associated with the proportion of phenomic characters? And our results using non-parametric experiment tests indicated that all topological metrics, so Robson folds, cluster information distance, and SPR moves, all of them were correlated with the proportion of phenomic characters. So the higher the number of phenomic characters relative to the number of molecular characters, the higher is the degree of incongruence between molecular and total evidence trees. The question 3 is the addition of phenomic evidence equally impactful on likelihood and parsimony topologies? Well, given we used here some Friedman tests to test this question, which takes into account the non-parametric distribution, the non-independence of the data, and here the Friedman test indicated that for cluster information distance, we have the values of parsimony analysis slightly higher than maximum likelihood. The same was not observed in Robson folds and SPR moves. So when we assessed the contribution of phenomics to the optimality score in total evidence trees, basically we computed the proportion of the optimality score from phenomic only trees divided by the optimality score of the total evidence trees. And we found that it can be lower or higher in parsimony. So in 29 data sets, it was higher in parsimony than likelihood, and 23 data sets, it was lower in parsimony than likelihood. And another important aspect here, the difference between the optimality criteria, is that the resolution, it's variable in parsimony analysis. We can keep, increase, or decrease the resolution, but not in likelihood, they were always binary. Oh, okay, so now the questions related with support values. The fourth question was, how do bootstrap values of shared clades differ between molecular and total evidence trees? In parsimony trees, bootstrap values are slightly higher than total evidence trees. In parsimony trees, the total evidence, the bootstrap values of total evidence trees are slightly higher than those from molecular trees. Specifically, the bootstrap values were the same for 43% of the nodes, higher in molecular trees in 20% of the nodes, and higher in total evidence trees in 36% of the nodes. In likelihood trees, there is no significant difference in bootstrap values between molecular and total evidence trees. So the last question was, can bootstrap from molecular clades retrodict the presence of total evidence? The clades, which is, can we use the bootstrap values from molecular trees to predict the presence of the same clades in total evidence trees? And yes, logistic regressions indicated that the bootstrap values in molecular trees are positively associated with the presence of the same clades in the total evidence trees. But there are some extreme exceptions, some extreme cases of molecular clades with 100% bootstrap values that are absent in the total evidence trees. So if you rely on 100% values, we shouldn't, because there are some cases that are exceptions. So some take-home messages, the addition of phenomic evidence can impact both topology and support values. In topology, phenomic impacts them, and the more phenomic data you have, the more impactful. And the impact is slightly higher in parsimony than likelihood, according to one of the topological metrics, the cluster information distance. And also, regarding the impact on support, we have support values slightly higher in parsimony analysis, in total evidence parsimony analysis than molecular parsimony analysis, but the values, the p-values are very close to 0.05. And for likelihood, we didn't find no significant results. And regarding if we can predict the presence of clades based on support values from molecular trees on the total evidence trees, yes, in general we can, but some extreme cases occur with the 100% values of the molecular clades that are not present in the total evidence trees. Okay, that's it. Thank you very much.
Great, thank you so
All
much.
right,
We
I think
have a minute or two for questions.
Okay.
I had a question. So in your total evidence analysis, how many fossil ages did you say you had?
Oh, we didn't use fossil because we didn't use fossils because we had the same composition of leaves in the molecular and the total evidence analysis. So we didn't include fossils in our preliminary result. We will include in the future. Because and the reason for that is because we want to be sure that our results here presented are related with the addition of phenomic data and not for differential taxon sampling. We don't want to confuse the things, the causes of these results.
Right. I think that's
Great.
about all the time we have for questions. Looks like Brian is ready. Brian, you are muted.
Year 18 of Zoom. Okay, this is a talk presented in collaboration with Jeremy Beaulieu, a long-term collaborator, on TIPFOG. I'd like to start with acknowledgements. First of all, NSF, which has given money for this work, and has given money for basically all my work from the time I was a grad student up. So I love NSF. I'll note that this is them at 75 years, and this is the proposal for them at 76 years. So I might be support from NSF in the future, but I still hope so. Also acknowledgements from for R, especially the targets package, members of our labs, Matt Hahn and 2 reviewers on this work, and also the organizers and the sponsoring societies. So for over 40 years, scientists have seen a pattern of faster evolutionary rates over shorter time periods. It's a hollow curve on a plot of rate versus time. It's a linear curve, often a slope of -1, on a log-log plot of log rate versus log time. And there's been recent work on this across molecular data, morphological data, diversification rates, fossil origin rates, and others, right? And there's speculation about this linking like micro and macro evolution. And so Jeremy and I have been looking at this pattern for over a decade now and sort of poking at it and wondering about it. One thing we're thinking about is what's a rate, right? And so a rate is a difference in some trait value, right? So like log number of species or log body size or things like that, divided by difference in time. We don't actually see the underlying true value, right? What we see is an estimate of the value. So it's the value plus some error term, epsilon, right? For example, are there exactly 973 species in a clade? Right? Is this tree species exactly 17 meters tall? Right? So there's some fuzziness there. And so by doing some algebra, we can get this, right? And we have basically 2 terms here, right? One is the desired process, right? One's the thing where we have difference over time. And our expectation is as time becomes shorter, right, that approaches 0, right? So you have things that are identical in time, they should be identical in, body size difference and things like that. We also have a noise process on top of that, right? And so if you think about like looking at squid length, right? So squids are squishy, right? So you're gonna have 2 species at the same time period and measuring them, right? Depending on how eager your assistant is in stretching the squid, you'll get different lengths of body, right? And so this process might be independent of time, right? Where you have a plus or minus 10% for the body length. It doesn't matter how far apart the things are in time, you still have each one has a plus or minus 10% value. And so you made a model to fit this, right? So one is a model for the noise process where you have some constant amount of variation, h, and then we divide that by time, right? And then we have a desired process, and for that we had a simple model of increase or decrease in rate and or a constant rate. When we fit this to those original data from Gingrich and found that most of the pattern, the pink one, is being driven by this noise process, right? You only start getting a linear curve at the very end, right? Those gold and blue areas. The green line shows the actual rate when you remove the noise process. And so rather than this nice, linear thing through time on a log-log plot, basically saying we're getting a hyperbolic curve from the noise. Another way of looking at this, and whether it's an actual thing, is to randomize the data, right? So since we have both rate and time, we can use that to get an estimate of the difference in x, right, which is rate times time, and then randomize that with respect to time difference and replot it again, right? And this is an idea back from Pearson, 1897, right? Who thought about the spurious correlations, right? This is an example of this potential process. And so here we see rep 0 is the original data, and that gray line there, and every other rep is randomizing the data. And so this is a data set of body size. And you see that the original data and the randomized data look almost exactly the same, right? And so this error process is driving it. And we can see this across different data sets, different kinds, right? Body size, diversification rates, and so forth. Right? I had a paper on this, and so you can play with it. And the basic story is, we have the randomized data look a lot like the real data, right? If you actually predict the rate from the real data or for the randomized data work better. So it's kind of a surprising result, right? And so this 40-year question seems to be driven largely by assuming that there's no error in our data, right? Even if we're measuring, protoceratops versus, triceratops across different time periods, right? We know those lengths exactly according to the model, right? Which we don't. And so where else can this matter? And so we think about a phylogeny like this, right? Again, we have, species A and B that have zero divergence time, right? We expect it to be actual difference, right? And we're not the first ones to think of this, of course. So lots of people have talked about these things. So this is some sort of other variance where the evolutionary process doesn't necessarily map to exactly what you measure in the real world, right? And so this is known by different names, depending on what model you're using, what kind of data you're using, your background, right? So one specific variances, one residual variation, phenotypic variation, measurement variance, measurement error. Okay? And so people have studied how to deal with this. So Kuhner and McGill had a paper over a decade ago looking at this in phylogeny inference, right? Because there you have sequencing error, right? So when you get your sequences back from the sequencer, it gets a little wonky at the tips, right? There's some error rate. And so there's an error with sequencing, right? And so how do you handle that? And so they fit a model where, the y-axis is distance from the true tree, right? So this is good, this is bad, right? And this is the amount of error that they set the program to assume, and here's the true value, right? 0.001. Right? It's a very small error rate, right? 0.1% error rate. And they found that, if you assumed no error, you were further from the truth than if you assumed the true value of error, right? And as a field, 10 years later, we are here, right? This is what we're still assuming for our error rate when we infer trees, okay? For comparative methods, this has been studied a lot. And so a really good analysis of this is by Silvestro et al. In 2015. I endorse this message, measurement error should always be incorporated, right? But as a field, are we dealing with this? No. And so here's an example from their paper. So they simulated data under a Brownian motion model and then tested to see if Brownian motion or an Ornstein-Uhlenbeck model fit better, right? And here you see that even at as little as like 10% error rate, right? So my tree height is 15 meters plus or minus 1.5 meters, right? You reject the true model over half the time, right? And again, even after this paper, we are here, right? Assuming no error. And so we're systematists, and part of our, the default for systematists is if you name something, you have power over it, right? So we want to name this because it has all these different causes, right? Sequencing error, plasticity, uncertainty, other kinds of errors, right? It has all these different names in literature. We'll just call it all tip fog, right? And the image of this is this neutral, right? So it's not necessarily an error, right? This phenotypic plasticity is not something's wrong, right? It's just there's this variation that's not incorporated in your evolutionary model. And so can we put this into the evolutionary model? And so we first did it with continuous data, right? So like height, body size, things of that nature. And so if we say the truth is these horizontal lines for rate, right? And note this is on a log scale, right? If you assume no measurement error, no tip fog, you get very, very ridiculously high rates, right? It should be a rate of 1, they're actually rates 10, right? Same thing in an Ornstein-Uhlenbeck model, right? So doing what we do normally gives you very, very bad estimates, okay? And these aren't huge values for measurement error, like this is 10%, 20%, right? Even at, 0.05%, it's still 0.5%, it's still pretty high, right? 5%. And then if we correct that, right, by putting in tip fog, we get these other estimates that are much better, right? And there's two ways to put in tip fog. One is you can measure a bunch of individuals in a species and use that to get an estimate of the variance of the tips and put that in. Another way is to estimate as a free parameter. As we've done both, the free parameter we call eta, right? Because we're running on Greek letters in comparative methods, so eta is good. And we see that it does pretty good job estimating for that. We can also do it for discrete states. And so discrete states, it's going from 0 to 1 and 1 to 0, and it's estimating those rates, right? And here the error isn't, height plus or minus 10%. Here the error is 10% of my 1s are actually 0s, and 10% of my 0s are actually 1s, right? So actually the data are wrong, right? And so this feels like it's a harder problem, And here we see if we assume zero tip fog, right, we get, again, on log scale, wacky rates, right, for 10% error, right? And you think about when you're measuring things, like you're measuring like woody versus herbaceous species, right? Okay, what's a liana? How do we characterize that? That could depend on who measures it, right? Is this a shrub or a tree, right? Is this a predator or an herbivore, right? There's fuzziness always in biology. No matter what, every time you discretize something, there's always fuzziness, right? And that can result in error, right? And so here we're trying to figure out the effect of that. And so again, if you ignore that error, that tip fog, you get the wrong answer. If you estimate it, you get a wrong answer, but less wrong. So that's nice. And here we see how well do we do estimating that tip fog rate. And here's the truth. Here's our estimate, and it's pretty good. All right, and these are not for giant trees either. All right. What about for diversification models, right? So like things like BISSY, HISSY, and FRIENDS. So we found that, and here again, it's there's concerns because you're trying to estimate like, okay, some of these data are wrong, some of these data are wrong. We're trying to estimate all these rates for these models, right? And we found that if you ignore tip fog and assume it's zero, you get bad estimates for transition rates. But if you incorporate it, it's good. If we care about diversification parameters, so here's net turnover rate, we find that actually has very little effect, which this surprised me. In some ways, things become more similar if you assume tip fog because model has trouble figuring out what's the true zero rate, what's the true one rate, since some of them are in the other category, right? But it's still not bad, and so it still gives you better results overall than ignoring that. And then identifiability is a huge concern for diversification methods with lots of good reason. And here we see, can we estimate eta tip fog in a BISSY HISSY model? And yes, we can. So if we simulate with this, we can still estimate it pretty well. So that's reassuring. So some caveats. So first, we model tip fog as a constant proportional error, right? So 10% plus or minus. You can instead use absolute error, right? So height plus or minus 1 meter, right? So you can see how those play out. You can mention other sources of error, right? So I'm trying to estimate, form for plants and trying to say like woody versus herbaceous. It could matter depending on who does the observation, right? Or the number of observations could matter, or things like that. And so we haven't incorporated yet that sort of variation in the tip fog. Tip fog doesn't yet address very large errors, right? So someone measured in centimeters, not millimeters, or someone flipped a sign in the latitude, or things like that, right? Those are big errors. That's not part of our model yet. Errors in taxonomy. So I have one species on the tree and I measure a different species, right? That causes an error. That's not incorporated in tip fog, really. We're also not incorporating things like noise and branch length or topology or presence or absence of reticulation or things like that. It's also a question of the best approach for dealing with the estimates, right? So if you can estimate, maybe you use a fixed value, like looking at measuring multiple individuals, right? For tree inference, where the given tree is not fixed, there can be identifiability issues depending on which models you use. If you use like Jukes Cantor model and a similar model for error, for a tip fog, you can't tell them apart. If you used a model like SELAC, that has like drift and selection, that is differentiable from tip fog. So it depends on which model you're using. That's something to explore more. So conclusions. Assuming every character has zero tip fog is a bad assumption. It's a very bad assumption, right? It's caused predictable misleading patterns, like the pattern of like rate over time for 40 years, right? So it should be the default, right? We know this has an issue. We know it's solvable. Solve it already, right? Like other people have argued, but we argue it again. Fixed value fog can be used if available. We've made it all available in our software, so AUI, Core, and HISSY. Other high-quality comparative methods programs have it. Not all of them do. For those who don't, add it. And it's not yet in a lot of pre-inference programs. And watch for our paper soon in Evolution. So questions?
Thank you. We have about 20 seconds left for a question. Let's see. And Robert, you're up next if you want to start getting your slides ready.
Yep, thank you.
You can go ahead whenever you're ready.
Okay. So So everybody see my screen? And can you guys see my mouse pointer? I'm going to assume the answer to that is yes, and continues.
Yes, we can.
Thanks everybody for coming. It's a real pleasure to be able to participate. I really like the online interface. You know, many of us hate airports, so thank goodness for that, for this online opportunity to talk. Okay, so there's my title. There's a fossil. The basic sort of question I want to discuss with all of you guys is this one. We have this problem in paleontology where the data that you know we like to use for reconstructing phylogenetics or phylogenetic trees are generally, those data are generally not available, with a few exceptions for very geologically recent cases. But you know, paleontologists look at Archaeopteryx. Generally speaking, this placement of Archaeopteryx is pretty widely accepted. It's among many other extinct species. It is regarded outside of the crown clade of birds. But you know, maybe this is just an artifact. There was actually a paper a little over a decade ago saying that fossilization systematically caused fossils to misleadingly appear at basal nodes. And I think it raises a serious question. How would we know? And what I want to tell you about in the next few minutes is this sort of heuristic method, artificial extinction. It's not new, but I have a particular application of it, and I and many other colleagues. The notion that fossilization itself causes species to misleadingly appear towards basal nodes, called stemward slippage. I don't think that's true, as I may have mentioned. If you want to read about that, here's the QR code to look at that paper Martin and Smith and I wrote a couple of years ago. The basic methodology for artificial extinction, though, is you take a living animal, like this shrew, and you pretend for a moment that it's a fossil. So what you're looking at the left is a phylogeny of only extant taxa, only recent taxa, all of which have lots and lots of molecular data available for them. You'll note there are some polytomies here. So you're what I shall call, for our purposes today, a well-corroborated tree. It doesn't have to be completely resolved, and as you see, this one is not. But given, you know, the number of possible bifurcating rooted or unrooted, for that matter, trees for N species, this is a teeny tiny little fraction of all the different possibilities. So artificial extinction takes our shrew, for example, deletes all of the DNA and other non-fossilizable data for that shrew, and then you rerun the analysis, including an anatomical matrix that sampled not only for that shrew, but also for these other living species. And I might add that the well-corroborated tree here is built entirely using non-fossilizable data in this case, right? So those are independent bodies of data. The anatomy on the one side that you can get from fossils, and the, generally speaking, the molecular data, the non-fossilizable data. Those are the data that are used to reconstruct this topology. Okay, so you reconstruct the topology with our artificial fossil, in this case, shrew, and then you measure the congruence with the resulting topology of that artificial extinction experiment. Does it look like the well-corroborated tree? And you can use all sorts of different measurements, shared splits, quartet similarity, Robinson folds. I think for this talk, I think I'm just going to share or use shared splits. We'll come back to that. Here is a slightly more detail on that point. So here we have a matrix. This happens to be in TNT format. We have a fossil Darwinius, a fossil primate, and like pretty much all fossils, there's going to be some part of comparative anatomy that wasn't preserved. Say there's no ankle bone that was ever known for Darwinius, and that's represented here by a bunch of question marks. We have a living taxon, the Dermopteran, right? The flying lemur from Southeast Asia. And if we're going to treat this as an artificial fossil, we retain, of course, the real anatomy of a flying lemur with all those zeros, twos, and ones here. We have a bunch of question marks, which corresponds to the fossil template here, and then the real character states over here. And of course, also the Dermopteran, you know, if we're treating it as a fossil, has no DNA. So we just replace all the DNA that would otherwise have had with question marks. And you go through each one of these living subject or recent subject taxa. You do a couple of New World monkeys here. Here's a lemur from Madagascar, et cetera. And you just do the same thing. Now, also in our method is a contrast with randomized character state. So in addition to replacing the missing data in a subject recent taxon corresponding to where those data are missing in a fossil, You can also get rid of the real character states and just replace them with random zeros and ones. And that comprises sort of a comparison that you can compare, contrast the real data with the randomized data. Okay, so I'm going to talk about one particular data set. I mean, I suppose, did I mention what that was in my title, I think, lipotyphlan, not an everyday term. That simply means shrew, hedgehog, mole, and Solenodon nisophontes from the Caribbean, a particular group of placental mammals. And this is a paper that Robert Haubo Yuan and Ephraim Dixon and Quentin Martinez, Patrick Arnold, and I published a few months ago. Please pardon the self-promotion. There's a QR code if you want to see that paper. We have in this paper a matrix of hard tissue fossilizable data for all of our recent species, moles, shrews, hedgehogs, Solenodons, plus Domnina as a fossil shrew, and a bunch of other fossils. Domnina, this early Oligocene shrew, is known for about just over 2/3 of its morphological data set. And this here is the time tree that we published. So taking into consideration a small molecular alignment, 19 kilobases, nuclear mitochondrial DNA, plus stratigraphic information, plus morphology. And this is what that tree looks like. This is the topology, the tree that we got from our alignment of molecular data, plus congruence with other published studies in the literature. So our morphological data, our fossilizable data, were not used to generate this topology. You see here, there are some polytomies. We don't really know for sure what the sister taxon of shrews are, and it's a bit uncertain what the root taxon should be. It doesn't matter in this case because those nodes are unresolved. And so this is the standard to which we measure our artificial fossilization experiment. So let me get to you, or convey to you what those results were. Again, here are the methods. We have 43 recent species, all except Nisophontes, a recently extinct Caribbean fossil, are living, but we have molecular data for Nisophontes, so that's in there too. We skipped the root taxon, so of the 44, did I say I meant 44 recent species, we just use 43 of them because we keep the root taxon the same for presentation purposes. We have 23 different fossils, so we use every possible combination of these 43 living species with 23 fossil templates. Domnina is our best known fossil, so 2/3 of its anatomical data are known. There's some very crappy fossils in here that are known from just a few tooth and jaw fragments, under 10% complete. You combine all combinations of 43 times 23, you get 989 analyses that are using the real character state. So the real character states, bumps on bones, number of teeth, etc., for a shrew, for a hedgehog, for a mole. And we use 989 randomized character states. In addition to that, we also explored, and this is different from previous applications of artificial fossilizations, we looked at treating multiple recent taxa in per analysis, right? So not just the single artificial fossil per analysis, but 2, 4, 8, 16, 32. The total number of phylogenetic analyses that we undertook was just under 12,000, 5,900 with real character states representing the real anatomy of these living species, and just under 6,000 of those are randomized. And because of the large number of phylogenetic analyses, you really need, you can't have any single analysis running for hours. It'll take years. So we need a really fast method. So we use TNT, parsimony using implied weights in TNT, and that way you can leave your analysis running overnight and get all your results. Okay, so let me tell you some of the specific results of this study. So here is a recent species, Ballerina brevicauda, short-tailed shrew, not rare in all sorts of parts of North America. We pretended, as I've been saying, that this was a fossil for a moment. So we deleted all the non-fossilizable data for Ballerina brevicauda. So we turned all of its molecular data into question marks, and we kept only the anatomical characters that were known in this particular case for Domnina. So 69% of 217, what is that, 250 or so. And where does it put Ballerina brevicauda on the resulting topology? Well, it does so here. And this is not far off, right? So in the well-corroborated tree, Ballerina falls out in a clade with Cryptotus. This is the one shrew that sneaks into the northern part of South America, that's where it should be. And it's not quite the same position in the artificial fossil experiment. In this case, it's the sister taxon of Blairinella, excuse me, but it's pretty close. It's certainly within the Sericine clade. And not only that, but reconstructed as the sister taxon to other sericines. So this is just anatomical data for Blairina that's putting it in that position. So that's not so bad. Now, when you randomize those anatomical character states for Blairina, this is what you get. It falls outside of the entire crown radiation of Sericidae. These are all the shrews. And this kind of is this fits to the older claim in the literature that when you delete non-fossilizable data, you get what they call stemward slippage. I mean, it kind of looks like that. Of course, this is just using random zeros and ones. So it's sort of a classic long branch. The branch leading to crowned shrews as opposed to other mammals is pretty long. And so that's where you get Blairina. But of course, remember, this is using random zeros and ones. It's not using the real anatomy that a real short-tailed shrew actually has in its teeth and skeleton, etc. Okay, so what about an example using multiple artificial fossils at once? Here is a result using real character states. Each one of these arrows points to a recent species. Here you see, what is that, 6 sericine shrews, so red-toothed shrews and 2 white-toothed shrews. All of these are using the template for Domnina, so 2/3 of 217 morphological characters. So using the real states of all these different living mammals. And again, it's not that far off. So you see Sorex, treating them as artificial fossils, Sorex minutus, Sorex coronatus, they both end up in the correct genus. Blairinella is not far off. It's there with our sister group to other sericines. It keeps the 2 crocodurians in the same place. I mean, this is not an identical topology. You can see Crocodura olivieri, for example, in the full data set is supposed to be the sister taxon of Crocodura russula. So it does change a little bit there. But in the grand scheme of things, this is not that horrible a result. The comparative anatomy is actually doing okay, putting these 8 different artificially fossilized recent species into this particular topology. Now, when you randomize those states, you get kind of a similar result as what I mentioned before. All of these randomized 8 artificial fossils glomp together and end up on what is probably the longest branch in this topology. Forgive me for just using a plain old cladogram here. But anyways, it's clearly a very imprecise, inaccurate result in terms of where these guys belong. And that's exactly what you'd expect with random data. So this is all for I guess, I have a self-interest in being a paleontologist myself, so I want these hard tissue data to work. But there it is. I think at least with this low bar of comparing random zeros and ones, the real data are actually doing not so poorly. Okay, so let me try and summarize all of these results in a couple complex slides. This one is slightly better than the one that comes next, which is my penultimate slide. But this particular slide shows you what happens across a bunch of different templates. So what I've showed you so far is just the results with Domnina, which is really a nice little fossil, 2/3 known for its comparative anatomy. The fossils get a lot worse, right? So here's our fossil with just under 10% complete. But the really good news for paleontologists is that as the fossils get more and more complete, the accuracy is here on the y-axis. These are shared splits with a grand total, I think, of out of 38 possible, you're going from just under 34 towards just under 36 as the x-axis gets more complete. So that's the amount of anatomical data that's represented by a given fossil template, right? This bottom part of the graph shows you the analyses treating 32 subjects in any single analysis. And you'll note the y-axis is a lot farther down, but that slope is still quite steep. In fact, it's actually steeper than what you get with just the one subject. So the overall accuracy goes down, which stands to reason with more artificial fossils. But
You've
this
got 30,
slope is pretty
30
good.
seconds
So
left.
as your fossil templates are getting more and more complete, the results are better and better. And this is an example of statistical consistency. You throw more data at a given problem, and your results get better, at least using our well-corroborated tree as a standard. Okay, so this is an even worse slide comparing the 1, 2, 4, 8, 16, 32 artificial fossils. And then you go, I think I'm just going to skip to my conclusion slide and tell everybody that the congruence with this independent, well-corroborated tree increases with the addition of data from fossils. And to read the rest, here's the QR code, and I'll thank everyone for their attention.
Yep, we've got to move on to the next talk. Do we have our next speaker here? You can pull up your slides.
So this is a, sorry, this is a recorded talk. So let me go ahead and pull up the video here. Try to full screen this.
Thank you everyone for staying for my talk, and I do apologize for not able to give the talk live. If you have questions, please do send those questions to my email address. The study I talk about today is recently published in Cisfile, so if you are interested in it, please do check the paper. And the paper is co-authored by Professor Craig Morris. The motivation of the study comes from the observation that have bothered me a while. The observation is that people tend to use species that are delimited by different criteria in the same phylogenetic analysis. So, for example, this case study used in our paper gives a typical example. We have different species complex in different color. We also have, together with the tip species in black, they are described by the traditional taxonomy, which means if we look at individual by its morphology, we are able to tell what kind of species it is. Now, as the technology of genome sequencing gets advanced, people are more often using genetic evidence to describe species. For example, this pair, and the purple pair has been recently elevated to a new species. I'll use the stars to represent the kind of evidence that people have used to elevate those lineages to species. The first stars means those lineages has accumulated substantial genetic divergence that they should be called different species according to the general lineage concept of species. The evidence could be coming from things like a structural analysis to show that there is no population admixture, or by testing or by comparing the model fit of with gene flow and without gene flow. Then people tend to add some ad hoc morphological analysis to show that there are statistically significant differences in the morphology between the two lineages. And note that those significant differences are usually quite subtle. It's different from the morphology in the traditional taxonomy. And for these differences, you can't tell which species it is just by looking at individual. You have to sample a large number of individuals from each population to detect those statistically significant differences. If we're lucky enough, sometimes we'll have pairs with contact zone. And if we have extensively sampled individual in the contact zone, we can reconstruct the deep genetic line across the genome at the contact zone. This provides strong evidence for some kind of mechanisms of reproductive isolation has evolved between the two lineages. So with those information, we were able to identify what I call the cryptic species, which is loosely defined as species that cannot be told apart by eye and are found in the same habitat, but have accumulated extensive amounts of genetic divergence between each other. So the question is, if we include all these species, describe the species in the same phylogenetic analysis, what does speciation rates actually mean when we describe different species in different ways? And more interestingly, if we want to test some theory on speciation, and if the prediction depends on what species criteria we use, then how can we tell those predictions apart? Another interesting question is, now using the genetic evidence, we tend to find lots of newly discovered lineages. For example, in this tree, we have sampled lineages in each species complex that have accumulated a substantial genetic divergence at the same level as those lineages that has been elevated to species level. So do we include those lineages in the same phylogenetic analysis, or do we exclude them simply because no one has ever elevated them into species yet? Well, neither way sounds reasonable to me. That's why we want to develop this new method to describe species in a probabilistic way. The idea is very simple. It's simply to use lineages with known criteria to estimate the rate of other lineages to evolving to meet the criteria, assuming that the rate stays constant across lineages in the same species group. So for example, we can estimate how fast do lineages become morphologically distinguished enough to be distinct morphological species by looking at the branching patterns that linking different morphological species. Or if there is any key diagnostic traits that can tell us when speciation completion happened. For example, males with blue throat color are preferred by blue females, which suggested that the evolution of blue throat in males would co-occur with assortative mating, which would drive the completion of speciation very, very fast. And lastly, we can use the branching pattern between cryptid species in order to estimate how fast do lineages become genetically diverse enough to be called cryptid species. Now, the idea is simple, but the mathematics is not that simple. I will try to explain it without too much mathematical detail. So the model is based on the projected speciation extinction process that we proposed 3 years ago. This model basically extends the projected birth death model from species level tree to lineage level tree. In the projected birth death model, the tree is species level because we sample one lineage per extant species. And the model starts with an initiation of speciation, which can be think of bifurcation along the tree. So now one species is bifurcating into 2 lineages. Both lineage would have the potential to become a distinctive species to its ancestral species. These lineages are called incipient state. And then it takes some time defined by the speciation completion rate lambda to transit from I state to G state, which G means a good species. So we can see this lineage through a speciation completion event transit into a new species, which leads to this good green species. Okay. Important slide, because we are working with species level tree, so the extinction rate of an incipient lineage is the extinction rate of one lineage, but the extinction rate of a good lineage means all the lineage of that good species have one extincted before the present. And that's why we have different extinction rates under different states. However, when we have sampled all the lineages in the tree, then all the lineage should have the same extinction rate because it's the extinction per lineage. We make this extension by replace the good state with the representative state. Importantly, the representative state is also for one lineage, which means regardless of the I state or R state, all the lineage should have the same evolutionary dynamic. The only reason that we still want to include some representative state is because we want to use the state to track how many distinct species there are at each time slice around the tree. For example, now we have 2 blue lineage of the blue species. One lineage must represent the species, and it actually doesn't matter which lineage we choose to represent this blue species. What matters is that we know the most recent common ancestor of each extant species of the blue species, orange species, green species has to be in representative state because we only got one most recent common ancestor as a single lineage here. And more importantly, for the orange species, which are paraphyletic here, this internal node must be an incipient lineage of the orange species. So this node is I, and its following node is R, which means using I and R state, we enforce at least one speciation completion event along this internal node. So in other words, following the number of distinct species at each time slice using R basically gives constraint that we have to have at least n-1 number of speciation completion events in order to have n distinct extant species at the present. And now we have trace each lineages independently, and all the lineage have the same evolutionary dynamic. It's very straightforward to expand the state and the policy model in order to account all different kinds of speciation delimitation criteria. For example, we have lambda C to account for the rate for lineages to meet the criteria of cryptid species. Lambda M would be the rate of lineage to meet the criteria of morphological species. And this Q matrix is the trait transition matrix. This is used to model any key diagnostic trait. And we also allow some proportion of the trait transition to not associate with speciation completion. For example, if we see the shape as the habitat, then event 1 is represent ecological speciation. This is when habitat transition co-occur with speciation completion. Well, event 2 corresponding to habitat expansion, where this incipient lineage of orange species changes habitat but does not change its species identity. We can also have event 3, which are speciation completion with some rate lambda, and we can allow lambda to change stochastically on the phylogeny. So the model is very general in incorporating different delineation criteria. So how well does PROSI estimate speciation completion rate? The height of the bar shows you the error between the estimated lambda and the true value of lambda used in simulation. The white bar includes all the simulated tree. The gray bar only includes the tree that have include at least one incipient lineage of a species. So it's amazing to see how much information just one incipient lineage can give you in terms of the upper bound of the speciation completion rate. And in general, if the speciation completion rate is smaller, then the differences between the speciation initiation rate and the extinction rate, then PROSI gives you a very good estimate. It's consistent and unbiased. Now I have talked about how we have extended PROSI to allow different speciation delimitation criteria, and I have showed you the accuracy of PROSI in estimating speciation completion rate. And in the context of this study is how fast a lineage evolved to meet a particular criteria. Then it's straightforward to use PROSI to delimitate species identity in a probabilistic way. So the approach we use is using a Gibbs sampling, which means in each MCMC, we sample the species identity of each lineage with question mark. And then conditional on those sampled identity, we ask what is the posterior probability of a particular lineage, for example, this north lineage of Carlia vivax belongs to, for example, the same species as Carlia vivax south lineage. So they are the same species. Conditional on the tree and the species identity of other lineages and the speciation completion rate, and also any traits that help us to estimate the rate of evolving to different criteria. This probability is simply the ratio between the likelihood calculated from our PROSI with the north lineage co-specific with the south lineage, and plus this likelihood marginalized over all possible species identity of the north lineage. And using this method, we calculate the posterior probability of those lineages which already have some evidence to show that they do not genetically admixture with any of the other lineages. And the posterior is quite high for those lineages to be distinct species. The idea of using lineages with known criteria to describe how fast other lineages can evolve to meet the criteria is not new. It's first proposed in this paper in this software called Delimitate. However, PROSI does have several advantages over Delimitate. Most important, Delimitate needs to list all possible configuration of speciation completion event in the phylogeny that is consistent with the current species identity of the tips, which means the RAM can be used up very quickly, almost in the order of 2 to the n, where big N is the number of lineages in the tree. Well, in PROSI, the RAM is basically constant time. And we have compared the power of both methods to correctly identify species identity in simulated lineages. PROSI has higher power than Delimitate over a wide range of conditions. But the most important advantage of using PROSI is that it not only delineates species boundary, but also can allow us to test predictions on different delimitation criteria simultaneously. So for example, in our study, we test this BDMI compatibility model, which I believe all of you know about it. Under this model, Gabriel's has predicted that the rate of evolving reproductive isolation is on order of mutation rates. And therefore, we, under a Bayesian framework, calculate the marginal likelihood of a model where the evolving rate to meet the criteria of quick-tip speciation is associated with mutation rate. And we compare that to the marginal likelihood of a model where the two rates are not associated. Unfortunately, because there's not much variation in substitution rate and among these lineages, we didn't find very substantial evidence for the BBMI model. We did find that the data favor the BBMI model, although not significantly. Because PROSI is such a flexible framework, that we have many more projects following up, and those are a list of the project that is currently going on. I'm running out of time to talk about each project in detail. If you're interested in any of them, please just email me and maybe we can figure out some collaboration to work together.
All right, thank you everybody for coming, and thank you for all our speakers. It was great learning about your research. That ends our first session. There'll be a 30-minute break, and our next sessions will start at 11.